Thank you for attending our Friday noon seminar. This is actually our last Friday noon seminar for the series. We'll be taking a break for the summer and we'll be back in September. However, let's welcome our speaker today. We have the pleasure of having Dr. Stephen Machtaler talk to us. He's an associate professor in the Department of Medical Imaging at the University of Saskatchewan. He obtained his PhD from UBC in cell biology. However, during his postdoc fellowship, he joined the lab in the molecular imaging program at Stanford University, studying cellular pathways as biomarkers for inflammation with ultrasound molecular imaging. And it looks like he's continued on with that work. His lab is currently focused on developing sustainable, clinically translatable microbubble for ultrasound molecular imaging of inflammation in humans, as well as fish for environmental monitoring. So please, Dr. Machtaler, take it away.
All right. Well, thank you so much for the intro. Hi, everyone. My name is, I'm just going to turn my camera off here for a second. But yeah, so my name is Steve Machtaler. I'm an associate professor here at USask. And today I'm going to be telling you a little bit about some of the work that we've been doing in my lab towards building a clinically translatable microbubble for ultrasound molecular imaging of inflammation for IBD. Sorry. All right. So today what I'm going to be talking about are what are microbubbles and how do they work? How do we detect them? How do we use them clinically? And then we're going to get into a little bit more of some of the research projects that we're doing in my lab about how we use them for molecular imaging and how we are working towards building one for clinical use. So first, let's talk a little bit about what are microbubbles. Now, these are actually, they're commonly used, especially in cardiology, but microbubbles are small vascular restricted contrast agents that consist of this protective shell surrounding a gas core. Now, these protective shells can consist of multiple different materials. The most common one right now is a phospholipid monolayer. And this here we have are the acyl chains. They are facing this hydrophobic gas that's in the center with the little head groups facing outwards. Now, this is the most common because these are the most echogenic, but they're also the most fragile and have the shortest half-life. We can make them out of other materials like proteins, like albumin, which is actually GE double optazon. It's made out of albumin. Or we can make them out of polymers. We've made some out of polyvinyl alcohol before, and these are kind of cool because even though their shells are really thick, whereas this one's about 3 nanometers, PVA shelled microbubbles are about 100 nanometers. They're so thick that we can actually use air as the contrast stage, or as the gas inside the middle, which makes them really economical. Unfortunately, because the shell is so thick, they're really bad ultrasonic contrast agents, and that's why nobody uses them. But it's really neat that we can make them out of different materials and different gases. Now, these gases tend to be these perfluorocarbon gases, which are high molecular weight, low solubility, biocompatible gases. But there's other gases that we can use. These are kind of the best in terms of how long a contrast agent lasts, but we can make them out of something like sulfur hexafluoride. So the important thing about microbubbles is that they're small enough to move through the microvasculature, but they're large enough to stay vascular restricted. So that means that when we inject them intravenously, they only stay within the vascular compartments and they do not leave there, which has advantages even in that it's a really nice, easy access to vascular imaging, but it also gives us a really direct access to our molecular target, which are actually going to be proteins expressed by vascular endothelial cells. And here, this is just a micrograph showing some of the microbubbles we've made in our lab. The big thing to note here is that it's actually a polydispersed population. So we get microbubbles of different sizes. So they generally tend to be less than 6 microns because the really big ones don't survive very long, but they can be submicron as well. So how do we detect them? So microbubbles are a really good contrast agent because we can actually detect them in multiple different ways. Now, the very first use of microbubbles was actually back in the 70s, and it was just a mixed up saline injected, and it was air bubbles. And may cause this enhancement of B mode contrast due to the change in acoustic impedance between the gas core and the surrounding fluid. So the acoustic wave is reflected off it. And this gives kind of like a traditional enhancement. So the B mode gets a little bit brighter, but it doesn't really work that well. But the cool thing about bubbles is that they're highly compressible. And so acoustic pulses can cause microbubbles to expand and contract asymmetrically. And it generates these unique acoustic signals that we can then detect with the transducers. So here's just a nice figure that shows that when you have our pressure wave, when it comes into contact with them, this is a region of high pressure, it causes compression of the microbubble, and they compress a little bit, and then they rapidly expand after that compression. And they can get quite large, depending on how hard you compress them. And so the other thing to note here is just this figure right here. This is a nice one, just kind of give you an idea of how microbubbles interact with these acoustic waves. This is a time radius graph. So if you draw a line through the center of a microbubble and you plot that over time, what you can see is that when it comes in contact with this region of high pressure, you get this compression, then they rapidly expand, then they compress again, then they rapidly expand. But here they can do this stably. So once we cause this expansion and contraction, the microbubble remains intact. Now we can also do this to be not stable. We can initiate inertial cavitation, and that has been used in different more therapeutic applications for, say, for instance, in a tumor, destroying some of the vasculature by hitting it so hard that it actually disrupts the blood vessels. Or we can do it really gently, cause this expansion and contraction. And this is actually used therapeutically for permeabilizing the blood-brain barrier. So they're doing a lot of this at Sunnybrook Hospital right now, where they're using MR-guided, high-intensity focused ultrasound to cause microbubbles to expand and contract, which causes permeabilization of the vasculature. And you can get drugs out. Super cool stuff. I'm not going to be talking about that today, but it's something, if you are interested, we can chat about later. So microbubbles generate a harmonic signal. So these are integers of the incident beam. And so if we take a look here, this is our waveform from the transducer, we can see that microbubbles expand and contract at different frequencies, which is key to how we actually started detecting them. So what you're looking at here, this is just a B mode image, and this is a tissue phantom where there's a tube running through it with some ultrasound microbubbles within it. And you can see that, yeah, we get this little bit of enhancement in B mode, but when we start to listen for these harmonic signals, we're starting to see this really nice, bright signal. And that was really good. It was a big advancement when it came out. The problem is it's pretty low sensitivity because the echoes are weak compared to tissue. Now, some companies are still using harmonic imaging, but they're combining multiple harmonics. And there's even a company in the States that they're actually using, like, I think the fifth harmonic to generate their contrast. They generate their contrast differently, but harmonic imaging isn't dead. It's just it's gone under some different changes. But the way that we mostly detect ultrasound contrast now is through this phase inversion imaging. So this was developed by Peter Burns at University of Toronto, where he used this nice, clever trick to increase the sensitivity of harmonic imaging. This is this phase inversion imaging. So tissue are linear reflectors. So they give, when we send in an acoustic pulse into a tissue, we get back an echo. Now, transducers spend only 1% of their time transmitting and about 99% of their time listening. So we send in a pulse, and then we listen back to the echo. Then we send in another little pulse of about five waves. And this is a phase inverted pulse. And so that when we get the echo back from the tissue, when we sum them, it should cancel out. And that's kind of our tissue signal. So when we put microbubbles into this, the microbubbles are nonlinear reflectors. And so the two inverted pulses actually give back different signals that when we sum them together, we can actually see the signal. And that's great because this allows us now, if we take a look at our little pictures here of our B mode, and this is our harmonic imaging. If we take a look at this pulse inversion imaging, now we can drop out all the background, which is fantastic, and we can just see where the contrast is. And we don't lose this data, so it really is the only imaging modality where we can get our contrast signal and our anatomical imaging simultaneously in real time. So it's a really cool modality. So you can image contrast in real time, and you can get some really important data from it. So let's take a look at how we currently are using this clinically. Well, we can use this right now for imaging vascular perfusion in real time. So the way that this works is we have our patients, we IV inject them with a bolus of microbubble. And what we do is we have the transducer over a region of interest, and we watch how the microbubbles wash in and wash out. And that gives us our really nice time intensity curve. And based on this time intensity curve, we can then fit different models to it. And then this gives us a whole bunch of different parameters that actually have clinical use. For instance, the most common one that we probably use clinically is peak enhancement, which is kind of a measure of vascularity in that region. Mainly vascularity and kind of like overall amount of blood in that region. So this is the peak amount of contrast you get in, but we can also get wash-in rates, things like this, blood volume, lots of different measures. But peak enhancement right now is the one that's most clinically used. So let's take a look at a couple of examples of what this looks like. Now, the first one is actually from my lab, and where we were using perfusion imaging to image fish liver in response to environmental toxins. So what you're looking at here, this is a B mode image actually of a rainbow trout. And it'll be its heart right over here, and this is the liver. And what we did is we imaged these trout before and after we gave it a dose of this drug, propranolol, which is a common used drug in humans, the vasodilator, and it slightly slows heart rate, but it ends up in wastewater, and it can actually affect fish. So we wanted to understand how it actually is affecting fish. So again, here's our B mode, and over here is our contrast mode. So this video is a little gimmicky sometimes, but let's just take a look here. So we inject our contrast, and if you look on the right, you can start to see it flowing in, and it starts to fill up the vascular compartment of the liver. So what we were able to do here is we draw a region of interest over the section of liver that we're interested in. Right here, and we plot that over time. And here you can see these two repeated doses that we gave ultrasound contrast, really similar perfusion curves. That was great. We administered this drug, propranolol, and then we administered the same dose of contrast. And because the vessels or the vascular space inside the liver started to dilate, we got more contrast in, and then we could measure this really nice change in contrast. And so this is the last time I'm going to be talking about fish, but it's actually a really cool application for contrast-enhanced ultrasound imaging for environmental monitoring, because it's non-lethal, which is a really important thing for environmental monitoring. But let's take a look at some human examples. And this is a lot of the work that Stephanie Wilson really pioneered out of University of Calgary in characterizing different liver tumors using contrast-enhanced ultrasound. So again, here's our contrast-enhanced ultrasound image of patients. This is their liver. And so we shouldn't see really any contrast here. We inject our contrast, and we see this nice flash filling right here. And then we start to see the flash filling up of the vascular compartments, and then this washout of contrast. Now, this isn't the greatest example, but it does show a couple things nicely. So what you can see here, if I just kind of move it along kind of slowly, this region right here, and this is actually where there's a metastatic colon cancer tumor, we get this flash filling of it before, say, other regions of the liver. And so we get this beautiful flash filling, and then over time, we start to see that the contrast inside starts to leave, whereas the contrast in the rest of the tumor still stays. Now, these tumors have really interesting wash in and wash out characteristics. And Stephanie went through and characterized a whole bunch of them in different tumors. And so based on the wash in, wash out criteria, she's been able to identify many different tumor types, just on those characteristics. So it's really interesting definitely worth taking a look at some of her work. Here's another example one, and I really like using this when I'm teaching ultrasound to the radiology residents, because it has this nice beam mode image here. And I ask them to take a look at it and tell me what they see. And they're just like, well, it looks like liver, right? But it's actually two large hemangiomas in here. And they're hard to pick out. But here's our contrast mode, and then here's our beam mode. And look what happens when we inject our contrast. All of a sudden, it's really easy to identify where those two hemangiomas are, right? You get this beautiful filling, super bright, fills up before the rest of the tissues. Really nice and easy. And here's another example of this is a different metastatic cancer. And I want you to just keep an eye out up here on this region. Now, it seems like this is maybe a second dose because there's a little bit of a background in this patient, but you can see when we inject the contrast, we get this. I'm just going to pause it right here. Right. You start to see this nice rim enhancement around the outside of it. Right. And then we get filling of the vascular compartment in the tumor or in the liver. And then this is gives us, I think, our best example of this enhanced washout. So again, you can see here, if this is the center of the tumor, all of a sudden, all of the contrast kind of just leaves. And we see this enhanced washout with this rim enhancement, which is really characteristic of liver metastases. So it's kind of a really interesting way that we can use real-time perfusion imaging to start to characterize liver cancers. Now, what we do in my lab is mainly focusing on ultrasound molecular imaging. Now, molecular imaging uses contrast agents to visualize biological processes instead of anatomy. Now, within regions of disease, the vascular changes and expresses unique proteins that are involved in the body's response to those diseases. So we can add, in this case, targeting agents like antibodies to the surface or of these microbubbles, allowing them to stick and highlight these regions of the vasculature. So by imaging these proteins, we can get a nice indication of disease location and activity. So let's just take a look at this poor guy. And he's got some sort of Staph aureus infection in his arm. Now, this causes activation of the resident immune cells, and those send out signals like TNF-alpha that interacts with vascular endothelial cells. And these cells then start to express proteins that are involved in the recruitment of immune cells into that spot to deal with it. Now, what we're really interested in my lab is imaging these proteins down here, the P and E selectin, which are involved in the early events of transendothelial migration. And so we design our microbubbles to stick to these targets, and it tells us where we have sites of immune cells leaving the vasculature and moving into tissue where there's inflammation. So it's a really nice approach to that visualizing inflammation because we use the immune system to tell us where the inflammation is. All right, so once we get our microbubbles there, we have our targeted microbubbles. How do we actually detect it? So let's take a look here. So here's our targeted bubble, and here's our ligand that it sticks to. And here's our ultrasound transducer. So we inject a bolus of microbubbles. Now, some are just going to whiz on past, and they aren't going to interact, but some will stick to and bind to these regions of interest, or these proteins of interest. So what we do is we inject a bolus of microbubbles, these targeted bubbles, then we wait for a few minutes. And this allows for some accumulation on the surface of the blood vessels, but we still have this blood pool fraction. So we take a couple images of microbubbles, which consist of the blood pool fraction and our bound fraction, and then we apply a destructive pulse. And then after that destructive pulse, we immediately image the reperfusion. And that gives us this difference. So we have our pre-microbubble destruction, which is bound and circulating microbubbles. and we destroy it, and then we image just what's left over, just the circulating microbubbles. And the difference between these two signals is how many microbubbles we had stuck, and this gives us our targeted signal. So the more microbubbles we have stuck, the larger our targeted signal is. Now, how well does it work? Well, this is actually an example that I used in my postdoc, where I developed this mouse model of acute on chronic inflammatory bowel disease. And one of these bowels has acute inflammation on top of this chronic background, and the other is quiescent, where it has no active inflammation, is actually in this healing phenotype. Now, I sent these to the pathologist, and they both came back with maximal inflammation, even though I knew only one of them had active inflammatory processes ongoing. Now, we asked whether ultrasound molecular imaging using these PNE selectins can actually distinguish between these two, and we were able to see that. So what you're looking at here, this is our targeted sonograph. This is on day 0 before we induced acute inflammation on this. So this is our kind of our the quiescent state. And you can see that after we induce the active inflammation, we see this nice enhancement, targeted enhancement, that actually dissipates over 5 days. Whereas our control, we actually didn't see. We saw a little bit of a signal, and we learned later that ethanol actually, which is our vehicle in this case, it actually causes a little bit of inflammation. But still, what you can see here is we have this really big signal difference between active and quiescent inflammation. So how do we actually translate this? So if we take a look here, so this is actually a microbubble that was made by a company, Bracco. And unfortunately, so it was a clinical grade contrast agent that they were prepping for clinical trials, and then they abandoned it. And they abandoned it because the targeting agent in this case was so incredibly expensive, they just couldn't make it reasonable. And they wanted to focus on another contrast agent that they had developed for imaging cancer. That's called BR55, which visualizes angiogenesis. So what we wanted to do, or doing in my lab, is really understand, like, we want to make a clinical grade microbubble, but we have to solve a lot of these problems. We need to come up with some chemistry that makes it easy to add targeting agents to the surface, and we'll talk about this a little bit. We need to use a targeting agent that's resistant to the stresses of microbubble synthesis. We need to get away from antibodies and really start moving into some other ones that can handle a little bit more of a harsh environment. We need to make it more cost effective, and then we need to develop a clinic ready formulation. So I'm going to talk about a few projects that we've been doing in my lab to address some of them. Now, ultrasound molecular imaging is actually really easy to do poorly. So let's just say you buy an ultrasound system, preclinical system, and you really want to get into ultrasound molecular imaging. Contact the company, and they'll sell you these what are called target-ready microbubbles. And what this does is it uses this, actually it's a beautiful reaction between streptavidin and biotin, where streptavidin and biotin, these are just biological proteins made by Streptococcus, and they interact and form a really, really strong bond together. And so it's commonly used in in molecular biology. It's really quick. You do it at physiological time points. The problem is it's immunogenic. And so you can't actually translate this. So if you come up with a successful targeting agent, say, oh, this works great, you have to then use a different ligation method and then redo all of your studies, because just changing this little, the way that you connect things to the microbubble changes biodistribution, it changes how your contrast agent works. So what we wanted to do is we wanted to develop a targeted microbubble that uses something that's just as easy as biotin streptavidin to add targeting agents to the surface, but is clinically translatable. And we focused on this strain-promoted azide alkyne reaction. This is copper-free click chemistry, basically. And we use this to test inflammation in a murine model of inflammatory bowel disease. And you'll see a lot of this model throughout the next few projects that we're going to be talking about that we did. So this work was done by student Una, who's just about to, she's just writing her thesis right now. We published this in 2022. So what we did here is we asked whether we could use copper-free click chemistry to add antibodies to the surface of our microbubbles. So we designed some microbubbles where we switched out, we added a lipid that had an azide on the end of a long polyethylene glycol chain. And then we labeled antibodies with these bifunctional linkers that allowed us to put on this DBCO group on the end. And the cool thing is, once we do all of this, all we do is we just add these two things together in saline, and they click on, and there we go. So we asked whether or not this approach works just as well as biodavidin. The only issue with that copper-free click chemistry approach is it still takes like 2 to 4 hours, which is similar to like NHS amide reactions, malamide thiol reactions. So we asked whether or not we could just do this a little quicker. Now, microbubbles, when we inject them, they go into from like a cooler room temperature to 37 degrees Celsius. And we know that they're stable at that temperature. So we asked if we could use this 37 degrees to speed up this chemical reaction. And what you're looking at here is a bar graph where we compared the fluorescent intensity of microbubbles with this azide alkyne reaction at different temperatures over time. And what we're able to show is by about 30 minutes at 37 degrees, these we're getting just as much labeling as we were at 2 hours at room temperature. And that's great, because if we can make it as quick and easy to use as biotin streptavidin, then I think that we can get more people using clinically translatable contrast agents. And just changing temperature, it didn't really have a change in the microbubble concentration or the microbubble diameter over an hour. So it didn't seem to affect the bubbles. And you can take a look at this micrograph here. This is our T-selectin targeted microbubble using our biotin streptavidin or azide DBCO. And you can see that with a secondary used to detect this antibody on the surface, we could see fluorescence on there. So it was great. So we were able to use azide alkyne reaction to click on an antibody onto the surface of these bubbles. But does it actually work if we try and image it? So we utilize this animal model of inflammatory bowel disease that we use in my lab all the time. It's this haptenizing agent called TMBS. So this is a picrolyl sulfonic acid. And what it does is it sticks to all of the intestinal contents, and we inject it with 50% ethanol. And that 50% ethanol actually permeabilizes the epithelial layer surrounding the gut lumen and allows these haptenized antigens to then pass into here and cause a T cell mediated inflammatory immune response. So it works really quick. We get an inflammatory response within 24 hours. It's repeatable and nice and easy. My students love it because it doesn't take weeks and weeks to initiate. And so you can see here with our saline control, compared to our TMBS, we have really nice hallmarks of inflammation. We get immune cell infiltration, edema. In this particular one, there's actually even a little area of necrosis. But more importantly, we see expression here. If you look at these micrographs in red, we see expression of T-selectin on these vascular endothelial cells. We have this nice dilation of the vessels as well. Great. That's what we're targeting. We're seeing expression. Can we actually see an ultrasound molecular imaging signal? On the top here, these are our biotin streptavidin microbubbles, and we were able to see a differential signal. It wasn't anything to write home about, but it was statistically different. And when we compared that to our azide alkyne, not only did we get a similar targeted signal, but the nice thing is that we saw a decrease in our background compared to this biotin streptavidin, which is really common with these biotin streptavidin bubbles. For some reason, we see a lot of non-specific sticking to the vasculature. So depending on how great your antibody is, it can be really hard to distinguish the two. Whereas with this approach, we saw no real background signal, which was excellent. So to conclude, we were able to use copper-free click chemistry to add an antibody to the surface of a microbubble. This can be done almost as quickly as the streptavidin biotin. And the results are comparable in ultrasound molecular imaging to streptavidin biotin with the same antibody. We were really happy about this because if we can start switching people from this biotin streptavidin approach to this DVCO azide approach, we can actually get them to skip this first step in clinical translation, which is, I think, really held back a lot of people from working with ultrasound microbubbles and clinically translating them, is because the first steps, it's really hard to really hone in on whether or not a particular target will work with these bubbles. So what we really want now is an alternative to antibodies. Now, why do we want an alternative to antibodies? Well, there's lots of different types of binding ligands, but most of them are based on these biological proteins. So we have antibodies, antibody fragments, single chain fragment, nanobodies, nautins. These are all biological proteins. And the problem with this is we have to grow them. So we identify them, we grow them in cells, we have to isolate them. That makes them really expensive. Not only expensive, but it also means that they're pretty fragile. So antibodies are sensitive to changes in pH. They're sensitive to freezing sometimes. We can't often apply heat to them because that'll denature them. So it really limits what we can do in terms of down the road packaging of a targeted microbubble. So we want to know if there are some other binding ligands that we could make use of that really had this high binding specificity and affinity, but were quick and easy to produce and had a reduced cost. So this led us down the road to these things called aptamers. Now, aptamers are short oligonucleotides that can be made out of DNA or RNA. And what they do is they fold into these 3D structures. And these have a high affinity and specificity for certain targets. And so you can select them just like you would with an antibody. However, they're really low cost because you can synthetically produce them. It's just like getting primers. They're 30 cents a base pair sort of deal. So they're really inexpensive to use. The thing that's really held them back, so they were really hot for a long time. But the problem is with traditional imaging approaches is that they're really quickly degraded by nucleases in the blood. And they're rapidly filtered via kidneys because they are so small. Like they're in general like 30 to 50 nucleotides long. So they quickly degrade and they rapidly are lost, unlike antibodies. But actually pairing it with ultrasound is kind of perfect because aptamers can be modified to protect it from this degradation. So what we do is we can fluorinate some of these nucleotides, which kind of stops these nucleases from sticking to it and getting rid of it. And once we bind to these giant microbubbles, it's no longer filtered via the kidneys. So it seems like this nice marriage. So we asked whether we can generate an aptamer functionalized microbubble for detecting bowel inflammation in this mirroring model of IBD. So this is work done by, again, my student Una, that we published just at the end of 2022 in molecular imaging and biology. So what we did here is we searched the literature. We found this really cool RNA aptamer that binds specifically to mouse p-selectin. So we wanted to know if we could actually first generate a microbubble that sticks to it, and if it's stable, and how easy is it to label. Now, the cool thing about when we purchase these aptamers is that the company, IDT, that we get them from, they can just put an azide on the end, which is great for us. So they just send us these azide functionalized aptamers, and we just switch it out. We have a DVCO on the surface of our microbubbles. And here we're again targeting the same target that we've been looking at, which is p-selectin, which is involved in the early events of immune cell trafficking across the endothelial membrane in a blood vessel. All right. So how do we do this? So what I'm going to be talking about, we call this like our two-pot synthesis, and you'll see why. So we prepare our vial of microbubbles, and we don't actually use a vortexer. I don't know. My student gave me the slide and put on a vortexer, but it sounds like a little paint shaker, and it mixes it back and forth. After we activate these microbubbles, then we have to wash them because when we activate them, we have our microbubbles in here, but we also have all these extra lipids available. So we have to get rid of those extra lipids because we don't want them binding to our aptamer that we're sticking in. So we wash them, resuspend it into two different vials. Then we add our aptamers, and then we heat them at 20 minutes, or for 20 minutes at 37. Then we place it back on ice. Now, this gives us these really nice ultrasound microbubbles, and we were able to show we were actually able to get them with a fluorophore side 3 on the end, as well as the azide, that we could get this really nice labeling on the surface of our microbubbles compared to our controls. And when we worked with my colleague Laura Curell out of University of Calgary, she was able to look at the acoustic spectra of these, and we compared it to current commercial ultrasound microbubble called Definity, which is a somewhat similar approach to synthesis, a couple of different lipids. But we were able to see that we actually had very similar acoustic profiles to Definity. So it looked like we were going to be able to actually identify it when we imaged it. So when we used our nice TNBS model again, we actually did this in valve C. We actually did it in the second animal model. I'll show you again after. But in all of these models, after we induce our acute inflammation, we saw this really nice inflammatory response, expression of P-selectin on the surface. That's great. All right, so what does it look like? Well, once we injected our aptamer functionalized microbubbles, what we saw is this nice increase in contrast compared to our non-targeted microbubbles. So if you take a look at the quantification over here, our aptamer P-selectin microbubbles, or APBs, we saw a really nice increase, and that was much higher than our non-targeted microbubbles on the surface. That was great. We were extremely excited about this result, but we also wanted to try it in a second animal model. And here we use this genetic model of inflammatory bowel disease, IL-10 knockout. So these are missing interleukin 10, which is a potent immunoregulator. And so these develop chronic inflammatory bowel disease. So we did some imaging with this. We actually had to kind of boost the inflammation along because these mice weren't really developing it here in our facility. And one of the problems was we were actually starting to see a lot of necrosis. So our ultrasound molecular imaging signal wasn't as high as we were hoping because we were actually losing a lot of our blood vessels. But we still were able to see an ultrasound molecular imaging signal in a second animal model. We were really excited about that. So it seems that our RNA or aptamers can be loaded onto microbubble. They allow binding to their molecular target, in this case, P-selectin. And this does work in multiple mouse strains, which is great. Now we wanted to ask if we could use a better aptamer. Now, the previous aptamer that we used was this RNA aptamer that bound specifically to P-selectin. It was kind of our first introduction to the world of aptamers. The problem with it is, when we were talking about the cost of contrast agents, once we start playing around with things like adding fluorines to these nucleotides, it starts to make it really expensive. So what we want to know is if we could use just a long DNA aptamer that actually didn't have any modifications on it. So this is susceptible to nucleases in the body. But what we think is that's not actually going to be a big problem. So when we are talking about labeling our microbubbles, we're actually getting, in this case, about 140,000 individual aptamers onto the surface of each individual microbubble. So if you can think about it, that gives us a really high avidity. Even if over 5 minutes, which is a really short imaging time, even if that we lost 20% of the aptamers on our surface, we'd still have over 100,000 aptamers on there. We'd still have enough to bind And so that what we think is if we just make a really inexpensive abdomen, we'll still be able to see it, but we'll be able to dramatically drop the price down. So here we used another one from the literature, this DNA aptamer. That the cool thing about this one is it binds to both P and E selectin, both human and murine, and it's unmodified. So we decided to see if we could take this aptamer, add on our azide, and use it for ultrasound molecular imaging. So again, we used our same approach where we prepared the vials, we wash them, resuspend them, we add our aptamer, and then heat it to allow that interaction or that chemical reaction. And then we put it on ice. And we saw this beautiful fluorescence on the surface. We're getting, again, about 140,000 aptamers per microbubble. And what we did here is we first tested it in vitro. So we coated plates in recombinant mouse and human P and E selectin. And look how many of these bubbles stick. And what you can see here on these micrographs is that we were able to get lots of microbubbles sticking to mouse and human P and E selectin. Whereas our scrambled aptamer, so remember we're using a DNA sequence. So if we scramble up that nucleotides in that sequence, it no longer has the same 3D conformation, so it shouldn't stick. That's why we call it scrambled. So our scrambled controls don't stick to these proteins. So it looked like we were going to be able to get binding when we tested it in vivo. So what did it look like? Well, we used our mouse model that we get this really nice acute inflammatory response. And again, we're able to see this time an even larger ultrasound molecular imaging signal compared to our non-targeted control in these. We were really excited about this. But this is on a preclinical system. Now here we're using this beautiful visual sonics, high resolution ultrasound imaging system, but it doesn't really work the same way as clinical systems. So if we want to clinically translate it, we have to be able to have it work on a clinical system with clinical transducers, which we can't really do in mice. So we wanted to know if this would work in a larger animal model. And the first one that we took a look at was using rabbits. Now we thought that it would work because rabbit P and E selectin does have quite a high similarity to human and mouse P and E selectin. So we thought that it should work. And so what we did is we got some rabbits, we induced inflammatory bowel disease in them using our TNBS model, and then we imaged them with a Philips IU-22. Using a 12 megahertz transducer. It's a really old ultrasound system, but it actually works really nicely for this. So here I'm going to show you a couple of videos of what it looks like. On the top here, this is our scrambled control. On the bottom, we have our P and E selected microbubble. And you can see that we got a really nice acoustic signal from it. This is actually the bowel right here. This is the control bowel. And what we did is we injected it. We waited our 4 minutes, we initiated our destructive flash, and we see after. So we were so happy with this data because what you can see here is with our control, after we initiated our destructive flash, we had the same signal before and after, which indicates we had no binding. And with our targeted microbubbles, what we saw here is a signal, we initiated our destruction, then we had a change. This indicated that this many microbubbles were actually sticking to the rabbit vasculature in the bowel, which correlated well with our histology. So we saw this nice influx of immune cells compared to our nice healthy tissue. And then when we quantified it, yeah, we saw a nice difference. So here we have our comparison of our B mode and our contrast mode here between our scabs and our peds. So again, yeah, we saw a nice difference, an increase in our ultrasound molecular imaging signal, but it wasn't a huge difference. So we decided to try a different ultrasound system, but we were also had the opportunity to take a look at using pigs. Now, pigs, when we eventually go down towards large animal models, we want to start working with pigs. So we thought this was a great time to give it a try. Again, P and E selectin are very similar between pigs and humans. So we felt that our contrast agent would work. And here we were using a new, actually Samsung ultrasound system that we were demoing, and it worked. It was a beautiful system. So I'm sorry, this is my student Una here, and she was so excited to be working with pigs, as you can tell. But what you're looking at here, these are our pigs that we induced acute inflammation in their bowel. And this is our non-targeted control microbubbles, and this is our targeted control bubbles. You see we got a beautiful increase in contrast that dissipated quite quickly. Now, this was in pigs. Pigs are actually notorious for having the pseudo-allergic response. So ultrasound contrast agents, and actually contrast agents in general, tend to clear quite quickly in them. But when we took a look at this and quantified it, again, we saw again this beautiful, so in this little region right here, you can see our pre-flash we had and post-flash compared to our control microbubbles. We had this really nice difference indicating that we had specific binding. So we're currently looking at moving this towards clinical translation. However, we still feel it's not quite ready. We still feel it's not quite ready for clinical translation. That's because of how we're making them. So we're currently making them by preparing the vial. We activate them, we wash them, resuspend them, add our targeting agent, heat it, place it on ice. This is time consuming, and working with the radiologists and all the technologists, I don't think that they'll be able to do it. It's hard enough to get them to use a non-targeted microbubble, which is a one step, as opposed to have someone actually prepare all of this and use it. So why can't we just add everything to one vial? Now, a conventional microbubble, when we make them, we have our lipids in there, we have our gas, we shake it really rapidly, but normally this is done at 4 degrees Celsius. And we only get about 60% of the phospholipids in this vial incorporated into the shell. So in our hands in my lab, we used to only get about 2 times 10 to the 9 microbubbles per ml, which sounds like a lot, but compared to like a clinical, like Definity's into the 1 times 10 to the 10 microbubbles per mil. So we're never able to reach that level. And so you can imagine if we had all of these lipids, if we had our lipids labeled with a targeting agent, and we're only getting about 40% or 60% of them into the shells, we're losing about 40% of the targeting agent we put in. That makes it really expensive. It's because we're just throwing away 40% of our antibody or whatever we want to add in there. So we started working with a collaborator in Taiwan to get this. They have this really interesting approach where they actually heat the microbubble of the lipid solution prior to activation. And this really goes against the current paradigm of like doing it cold. However, what it does is it enhances liposome formation and greatly enhances microbubble production. So when we started testing this out, what we saw is that we're actually getting an order of magnitude more microbubbles. Actually, it became a problem for us because we're injecting contrast into mice, and all of a sudden we're dealing with an order of magnitude more. So we're having to try and pull out like 5-6 microliters out of a vial of microbubbles for injecting. But it was great for this because now 80 to 90% of the lipids are incorporated into these shells. So we're not actually losing that many microbubbles or of our targeting agent. And we find that it's really consistent. Like when we do this, these are 3 independent experiments. We see all the time about 2 times 10 to the 10 microbubbles per mil with a very similar size distribution using this approach. So what happens now? Can we actually just make a solution where we prepare our vials, add in our aptamers, let the reaction occur, heat them, and then activate them? So we skip all of these washing steps. Does it actually work? Well, this is what it looks like. So here's our pre-activation. So you can see this nice clear fluid, and our aptamer here in this case is Psi3, so it fluoresces red. And then after we activate it, you can see how cloudy it is. These are all the ultrasound microbubbles. And we see that even if we do this, we're still getting about 60,000 aptamers per microbubble, which is it theoretically it should be more than enough to actually stick to. However, this approach, because we get rid of all the extra lipids, we get way more aptamer on it, but we don't really know how much we need. So we asked whether or not this would be enough to stick on there. So in vitro, sticking to our recombinant protein, what we see is, yeah, it sticks. We can get it to stick to murine P and E selectin. Human P selectin didn't, for some reason, wasn't sticking so well to the human E selectin, but it was pretty consistent. And then when we image this, so these are the projects that we currently have ongoing. So we haven't finished data collection, but in a couple or two mice that we have, we were still able to get a really nice targeted ultrasound molecular imaging signal with these two mice compared to our control. So it seems that even though we're getting less aptamer onto our surface, if we make this one pot formulation where we just add everything and activate it, we can do molecular imaging. And then all we have to do is give that to the clinician. They just heat it up, activate it, and there they go. They're ready for imaging. So to conclude, we've constructed this one pot aptamer. This binds to recombinant protein. We see an in vivo signal in mice. And we really do think this is how we're going to get targeted contrast agents into the clinic, because all the clinician would have to do, or the technologist, is take this and then activate it, and then they're able to image. So with that, I'm happy to take any questions for the last 5 or so minutes. Yeah, and so I'd like to thank, especially this is most of the work, my graduate student, Una, who's just finishing her PhD and actually started a postdoc without finishing her thesis. So she'll be coming back for that. And then our collaborators, then our funding. So thanks, everyone.
Super. Thank you so much. Very interesting talk. Talks like this always reinforce the fact that I don't know much about biochemistry, but again, super interesting. Just the methodologies that you were coming up with were really great. We have time for questions. As always, you feel free to unmute and ask your question, or type it into the chat and I'll read it out loud. Let's go from the classroom in the Glen first.
Hi, can you hear me from here?
Yeah.
Yeah.
Thank you for the talk, Steve. Very interesting. I admit my biochem is quite rusty as well, but I have some physics questions for you. First of all, at what pressure is the gas in the bubble held, and how does it not explode or implode with the changing pressure of a blood vessel?
That's a good question. And a physics colleague of mine asked that before too. And I can't remember the actual class pressure in the bubble, sorry. But they do remain stable at that low shell. So the lipid shell actually reduces the surface tension and helps stabilize the microbubble itself. I would have to get the actual numbers for you. That was a good question. So, but yeah, they are stable, and actually people are making even smaller bubbles, which doesn't seem like it should work, but they do. These nano bubbles, it's a controversial topic, because there is a school of thought that some of them, that ultrasound systems can't detect them and some can. But yeah, I would have to get that actual number for you. Sorry, I don't have that off the top of my head.
Sounds good. And when the bubble fluid explodes, is there any biomechanism to clear the blood vessel of that lipid, or does it just kind of like accumulate in the vessels?
No one has actually followed the life or what happens to the lipids. It's such a small amount that we're actually adding. I'm guessing it just gets incorporated, so what happens is, if you actually watch them, when they start to oscillate, they actually fragment off and they form these little tiny, essentially like vesicles that can go into the bloodstream. But depending on how hard you hit it, you can actually drive them into the membranes of the vascular endothelial cells. And you can actually use that for like gene delivery. So there's this whole thing of sonoporation, where they coat the surface in a cationic lipid, put DNA on it, and drive these plasmids into the vascular endothelial cells for gene delivery. So we can use the fact that the lipids can actually move into the cells to our advantage. But yeah, that's what happens. But in general, it's such a small amount of material that we're adding. Like we can identify a single microbubble in a human using ultrasound. So we're not actually putting a lot of material in. But no one's actually radio labeled them and followed the actual where they go. So
Very nice. Thank you.
Thanks.
sure.
Morgan, go ahead.
Thanks for the really nice talk. I was wondering, so I know there's been some discussion about using microbubbles for more targeted chemotherapy delivery. And I think, like, at least as far as I've heard, a lot of this centers around like essentially popping the bubbles using ultrasound in the vicinity of the tumor. But has there been work done on targeting, say, like cancer cells specifically using similar techniques to yours, or would your technique be applicable for that kind of targeting of like actual therapy instead of just molecular imaging?
So the thing to remember, microbubbles remain vascular restricted. So they don't actually leave the vasculature because they're too big. So that a lot of the people that have been working on these nanobubbles, so Agna Exter, for instance, in Canada here, she's doing a lot of nanobubble work. There seems to be a debate in the field about whether or not you can physically detect them with ultrasound, but the idea here is you make really, really, really small bubbles, like in the 300 to 400, and you make use of EPR, so the enhanced permeability and retention effect, to get them out of the vasculature and into the actual tumor. But the problem there is you can't do molecular imaging like that because there'd be no way to wash it away. But you could theoretically load the drug and then cause them to release their payload. But it can't really get out. Microbubbles, the ones that we use, they can't leave the vasculature. So you'd be looking at spatial delivery just in that particular region, but we can spatially restrict where we do the delivery because that's where we put the transducer. So you put it physically over a tumor and then you just pop everything that flows through. So that's kind of how the idea of it right now is working.
And so that like the size of the microbubbles that like they still can't leave the vasculature even in something like a tumor microenvironment where you have kind of like your vasculature?
No, they can't. So the thing too to remember is, like, even though the gaps between blood vessels is quite large, like once you get out into the tumor too, like there's a lot of cellular material there. So we had a paper where we were trying to, or back in my postdoc, we were using like 100 nanometer nanoparticles, and we were using pretty high pressure to deliver them across the vasculature, and we could only get them moving in like 50 microns from the blood vessels, 50 to 100 microns. So it's what you even get out of the vasculature. It's hard to move large things through it. So a lot of the focus has been on just delivering the chemotherapeutics to that area, try and get a higher concentration at that site and a lower systemic concentration, or using some of these cool bioeffects where we can actually cause vascular collapse and destruction with really high pressures. You can do lithotripsy in addition to it, so we can liquidate all sorts of different things we can do with ultrasound. But that's kind of how the drug delivery goes. So we can't get it out of the vasculature. We can only deliver high concentrations to the blood vessels at that site.
Okay, thank you.
Super. I had a question myself. Your work with aptamers, I think you've demonstrated pretty well that there is some targeting when you look at the before-after destruction of the microbubbles. Is the idea for this to be a clinical translation, to be like a binary diagnosis, like we see a difference or not, or is the intensity, like the signal difference you see, is that another piece of information that can be used? I mean, or is it still too early?
I mean, the way we see it, or the way I see it, is this is a good tool for if you know where disease is and you have a specific question about it, this is the tool to use. And it can maybe be better to stratify patients into like needing PET imaging, for instance, or not needing PET imaging,
Okay.
or monitoring therapy. So, like, all right, with the inflammatory bowel disease, one of the projects that I'm really interested in is, can we use ultrasound molecular imaging to determine if the current therapeutics are working? Because like right now in IBD, like you give them a bunch of drugs and like, oh, okay, you feel a bit better. That's great. We think that the drug is working, but we have no quantitative measure to see like, oh, we see an actual quantifiable change in the inflammatory response. And that's why a lot of these patients, we still have like 30% of Crohn's patients requiring bowel resection within 10 years of surgery or of a diagnosis. Is like we have all these drugs and we don't know if they work because from patient to patient, how they interpret the pain is different. So they can be completely asymptomatic and have raging disease, or they may have just a little bit of disease and have huge symptoms.
Hmm.
So, like in terms of monitoring, that's what we would think. And then, like in terms of cancer, I think a lot of it is like you see a strange lump. For instance, can you use this to determine if it's like a benign or active tumor by looking, say, at active angiogenesis? Is it actively growing new blood vessels? So that's kind of, but I see it as kind of this in between, like PET imaging and like non-molecular imaging, and that it can really start to answer some of these like day-to-day questions. But I can't tell you if it's like, oh, you got, like a triple negative breast cancer sort of thing.
Sure.
Yeah.
Plus, I mean, it's also nice because it is, if you're using ultrasound, it's a non-ionizing radiation source. So there's always,
Yep.
It's always nice to be using something that's not giving dose to the patients.
Yeah. And if we can get it down to about $100 a vial, then it's a very different story. Then it's not a like an economic barrier to using it, and then we can ship them everywhere. So, for instance, in Saskatchewan, we can send them to northern Saskatchewan to a lot of these remote communities, and they have the ability to do molecular imaging there without being in a big center. So,
Right. Very nice. All right. Any other questions for Dr. Machtaler? If not, I'd like to thank you all once again. Thank you, Dr. Machtaler, for speaking to us. And we'll see you back here in September. Have a good summer.
all right. Great. Thanks everyone.
Bye-bye.
Okay. Bye.